Embryonic Editing via Mitochondrial Transfer by Hannah Lavelle. Approximately 1 in every 4,300 individuals in the United States have a mitochondrial disease. Inherited mitochondrial DNA mutations are transmitted maternally, and preventing transmission of these diseases is a priority for families. Some of the most common diseases related to a mitochondrial defect include Leigh syndrome, Pearson syndrome, and mitochondrial encephalomyopathy. However, there are also secondary illnesses that can arise with improperly functioning mitochondria. These can include diabetes, heart disease, Alzheimer's disease, Parkinson's disease, and muscular dystrophy, to name a few. An important new approach is a novel in vitro fertilization technique called mitochondrial donation, using either maternal spindle transfer or pronuclear transfer. Mitochondrial transfer works by replacing the damaged mitochondria in the mother's egg with healthy mitochondria from another woman's donor egg. The developing embryo now has nuclear DNA from the mother and father, as well as mitochondrial DNA from the donor egg. The complete human mitochondrial genome, made up of only 16,569 base pairs, was first sequenced in 1981. Clark and Shea pioneered the artificial mitochondria transfer, also known as AMT, in 1982, which involved transferring mitochondria with antibiotic-resistant genes into sensitive cells, therefore enabling them to survive in a selective medium and opening this new field of research. Around the same time, in an entirely unrelated area of research, the technique of nuclear transplantation between mouse embryos was described. The first human pathogenic mitochondrial DNA mutations were identified in 1988, and by 1995, the possibility of preventing the transmission of mitochondrial DNA disease by nuclear transplantation was already being considered. Since the work of Clark and Shea, the process of artificial transfer has and continues to mimic aspects of naturally occurring cell transport, especially in the mechanism cells naturally use to rescue other damaged cells. The AMT restores and increases respiration and proliferation and completes other cellular processes. These mitochondrial disorders can be devastating for couples trying to conceive. For example, one couple experienced several miscarriages and lost 2 children under the age of 7 to Leigh syndrome, a mitochondrial disorder that causes severe neurodegeneration. While the mother was a carrier for the disease, she did not have neurological symptoms herself because only a fraction of the mitochondria in her cells carried damaged DNA. Determined to have a child, the couple turned to Dr. John Zhang, an infertility expert at the New Hope Fertility Center in New York City. For the first time in humans, Dr. Zhang and his team attempted to use technology that has been around since 1983, mitochondrial transfer. In the case of his patient, this procedure resulted in the birth of a son without Leigh syndrome, seemingly curing an incurable disease. This first child was referred to in the press as a 3-parent baby when he was born in Mexico in 2016. This term is slightly misleading, as the child does not have an equal proportion of DNA from each parent. Rather, the majority of the child's DNA is from his parents, with only a small fraction coming from the mitochondria of the donor egg or third parent. The need to regulate the use of human embryos in both fertility treatment and scientific research was recognized in the UK following the birth of the first IVF baby in 1978. This major breakthrough led to concerns about the social and legal implications of such advances in human-assisted reproduction. To address these issues, the government established the Committee of Inquiry into Human Fertilization and Embryology, chaired by the now Baroness Mary Warnock. The report, published in 1984, set out a blueprint for the regulation of both IVF and embryo research, and with admirable foresight, included a chapter describing possible future developments in embryo research. This report was followed by the white paper, quote, Human Fertilization and Embryology, a Framework for Legislation, unquote, which was published in 1987 and formed the basis for the Human Fertilization and Embryology Act of 1990, also known as the 1990 Act. In May 2004, the team at Newcastle University applied for a research license from the HFEA to conduct research into using pronuclear transfer to avoid the transmission of mitochondrial disease. After extensive review of both the scientific methodology and the legal implications of this proposal by the HFEA, the team was granted a license in September 2005. In November 2010, the government was invited to consider exercising the regulation, making power added to the 1990 Act in order to make it possible for mitochondrial donation to be used as a clinical treatment. The 4th of March 2015 saw a historic event. The legislation was put in place to make mitochondrial donation legal in the UK. Critics of the technique have claimed the new law was rushed through and that there had been insufficient time to debate the issues. On 15 December 2016, the UK Human Fertilization and Embryology Authority, also known as the HFEA, announced that it would allow clinics to apply for licenses to conduct limited trials of the technique, which aims to prevent mothers from passing down mutations in cellular structures called mitochondria. The HFEA Scientific Advisory Board recommended that trials go ahead. Researchers at Newcastle University and the Newcastle Fertility Center have already said that they will apply for a license to carry out the procedure. Director of the Wellcome Trust Center for Mitochondrial Research at Newcastle University says that, quote, up to 25, unquote, selected patients could be treated at Newcastle with the therapy each year. The HFEA will approve each use on a case-by-case basis. After 17 years of extensive debate and discussion, mitochondrial donation may now, for the first time, give some women with mitochondrial DNA disease the opportunity to have a healthy child that is genetically related to both parents. While the UK is the first to legislate in this area, there is likely to be interest from other countries in mitochondrial donation. For example, the U.S. Food and Drug Administration has requested that the Institute of Medicine produce a consensus report into the ethical and social policy considerations of novel techniques for prevention of maternal transmission of mitochondrial DNA diseases.